welcome to another lecture. This lecture will be about the lymphoid or immune system. A lymphoid system is composed of lymphoid organs, of course. We divide lymphoid organs into central and peripheral ones. Central lymphoid organs are bone marrow, as you already know. All immune cells are made in bone marrow. And thymus is also central lymphoid organs, and that's also for gut or gut associated lymphatic tissue. And these are central lymphoid organs because immune cells gain immunocompetence here. So you already know from the lecture about blood that thymus has T cells, T lymphocytes, and they gain immunocompetence here in thymus, and other cells gain immunocompetence here in the bone marrow. And gaining immunocompetence means that they build up several receptors into their membrane. So for example, T lymphocytes build up T cell receptors in their membrane, so they're gaining immunocompetence or ability to react some antigens. Then we have peripheral lymphoid organs such as lymph nodes and spleen. They are encapsulated, so they have a capsule around them. Then we differentiate partially encapsulated organs such as tonsils. So they have capsule only on some side. They are not encapsulated completely, so only partially encapsulated organs. And also there can be free lymphatic follicles in the tissue somewhere. So these like small organs, really small ones, because there is only accumulation of follicles, so that's just like lymphatic tissue in some kind of wall of intestine or in airways. So these lymphoid organs are without capsule. Okay. Then lymphatic vessels are also part of lymphoid system. They start as blind capillaries and they merge into lymphatic vessels. What's really happening is that, we have some capillaries, so arterioles branch into smaller and smaller vessels, and then we have capillaries. And then what's going on, the fluid from the blood will filtrate outside to this interstitial space here. So fluid over here is called now interstitial fluid. So we have less blood here in the venules or veins right here, and the capillaries over here. So interstitial fluid needs to be put back to the system, And that's happening via lymphatic vessels and lymphatic capillaries that starts blindly somewhere here in this space, in this interstitial space. And this interstitial fluid is going to these lymphatic capillaries, and these lymphatic capillaries merge together to create the lymphatic vessels, and they put this fluid, which is now called a lymph, inside the lymphatic vessels back to the venous circulation. So what's happening? It's in this picture. You can really see it in this picture. There are many lymphatic vessels. They merge together to create two main lymphatic ducts, and that is a thoracic duct, which is on the left, and a right lymphatic duct. These are two main lymphatic ducts which put back this lymph back to the venous system in the places called angulus, venous, dexter, and sinister. This is more about anatomy, but you should know these two basic ducts that were created by lymphatic vessels merging together. So let's start with normal lymphatic capillaries. How do they look like? They are very similar to normal blood capillaries, so they are lined with endothelial cells, as you can see right here in this picture, normal endothelial cells. But they don't have tight junctions between them. So no tight junctions here, nothing here. So they are very loose. And also basal lamina under endothelial cells is discontinuous. So we have only some parts of basal lamina over here. So these lymphatic capillaries are very similar to sinusoids. Yeah, as if you remember, sinusoids also loose dilated capillaries. So they are very similar to them, and they have, sometimes we can see anchoring fibrils, which are anchoring lymphatic capillaries surrounding tissue. So this is how normal lymphatic capillary looks like. They merge into bigger lymphatic vessels, as we can see right here. The wall of lymphatic vessel is very similar to a vein. So they are dilated with thin wall as veins. This is the lumen. So it's more of like longitudinal section of a lymphatic vessel. And we can also see valve here. Valves are more frequent in lymphatic vessels than in veins. Yeah, so they are more frequent. We have high number of valves here in lymphatic vessels. So this is, once again, as I told you before, two main ducts that put lymph back to the venous system, because we don't want to lose fluid all the time. Okay, so we need to put back the fluid back to the system. But you should know that the lymph that is in the interstitium, in the space between cells in the tissue, needs to pass through at least one lymph node to get back to the venous system. So there should be always at least one lymph node in this passage. So, what's the general structure and classification of lymphoid organs? You know right now that we divide lymphoid organs into central and peripheral ones. There is also difference in supporting structure here, and you should be able to describe supporting structure, because sometimes it really looks similar in the slides, in microscopy. So sometimes you're not able to tell, okay, so this is a reticular epithelium and this is a reticular connective tissue. You probably won't be able to do that. But you should know that supporting structure in thymus is reticular epithelium. So reticular epithelium is for thymus, and reticular connective tissue is everywhere else. So also we can find reticular connective tissue in the bone marrow, in the lymph nodes, in spleen and tonsils, and so on. So reticular epithelium is only in the thymus, and that's very important because you should know that thymus is made of epithelial cells. Okay, and also there is not only supporting structure, because this structure is just supporting something that is even more important in that organ, and that are free cells. So we can find lymphocytes and their precursors and stimulated forms inside this organ, many macrophages, antigen presenting cells which are presenting antigens there to build up immunocompetency here, or to fight infection, or to simulate some immune response. So free cells are very important in these lymphoid organs. So we recognize two partially overlapping lines of defense, the innate immunity and adaptive immunity. You know this from the previous lecture probably. The first type is non-specific, and it's evolutionary older as adaptive immunity, and these cells are born like they want to fight infection. They don't need to acquire some kind of immunocompetence because they already know what to fight, what to eat, there are phagocytes like neutrophils and also eosinophils, basophils, right here macrophages, and also epithelial barriers are part of innate immunity, like your skin, but also mucosa in a gut or gastrointestinal tract or airways, also the mucosa. And there are also NK cells, which are basically lymphocytes, one type of lymphocytes. They are also part of innate immunity. They don't need to acquire immunocompetence or something else. And also complement system. Complement system is part of humoral immunity. Complement really means that there are many proteins together creating some kind of signaling pathway, or they are chemoattractants. They sign some, for example, bacteria or other immune cells. They are like markers for immune cells to do something. You will learn about complement system more in immunology. There are many types of complement pathways, and each pathway is composed of different proteins and different cascades. So they are activating and deactivating each other. It's very complicated, I can tell you. So, and adaptive immunity, we differentiate also like cells there, which are B lymphocytes and T lymphocytes. They are very important for adaptive immunity. And humoral part is represented by antibodies over here. Antibodies are very specific proteins which can bind to some antigens. Antigens is in general is a substance that generates an immune response. Antigens. So these antibodies can bind to antigens and block them or sign them for immune cells. So antibodies are important for humoral adaptive immunity. So just a brief recap. This is a lymphocyte, as you know. These are erythrocytes around, and this one is very small lymphocyte. We don't know if it's a B, L or NK cell. We can't really tell, but it's a lymphocyte, very small. This one is bigger, a medium-sized lymphocyte over here. So this one is probably more active because it's more cytoplasm here and lighter nucleus right here, so probably more active lymphocyte actually doing something, not just floating around. This is a thrombocyte over here, maybe two thrombocytes together. This is a picture from electron microscope. You can see the nucleus isn't quite spherical. There is some kind of indentation right here. We can see organelles here like mitochondria, and there is a centriole over here, and this is quite small magnification, so we can't really tell many other structures, but the cytoplasm here is basophilic for sure. We can see it here in light microscopy because it contains a lot of ribosomes and rough endoplasmic reticulum. Here I can see Golgi complex, also it's labeled with G, so I'm sure it's a Golgi complex over here. This is another picture, and what we can see here in this picture are these structures. I hope you remember these are the azurophilic granules over here. Azurophilic granules. Because lymphocytes are agranulocytes, but they do contain azurophilic granules. So we can't find specific granules, but they do contain azurophilic ones. Okay, so how do we differentiate lymphocytes? All lymphocytes are made in the bone marrow from a precursor cell called lymphoblast in the bone marrow, And based on the place of maturation, we divide B lymphocytes, which acquire immunocompetence in bone marrow and also in secondary lymphoid organs, but basic receptors are built in the bone marrow, and T lymphocytes, which acquire immunocompetence in the thymus, which is a central lymphoid organ, the thymus, and also NK cells, natural killer cells, which are part of innate immunity. So these two are part of acquired immunity. NK cells are part of innate immunity. So we differentiate many types of lymphocytes, as you know. B lymphocytes are produced in the bone marrow, and what are they doing? They go to a lymph node or to the spleen. They need to find some kind of follicle, so they are in the follicles. I'm going to show you how follicle looks like. So they are in the follicles. They're building these round structures in a lymphatic tissue, and they need to be transformed into the plasma cells to produce antibodies. So B lymphocytes will transfer themselves into plasma cells to produce antibodies, these proteins, which will now bind to some antigens. And this is happening in a lymphatic follicle. Next we have T lymphocytes, and we can differentiate many types of T lymphocytes, but you should know at least the cytotoxic T lymphocyte and helper T lymphocyte over here. Helper T lymphocyte. Cytotoxic T lymphocyte, for example, I'm going to have cytotoxic lymphocyte over here, has basic receptor as TCR receptor or T cell receptor on its surface, and all lymphocytes, all T lymphocytes have also CD3. T lymphocyte, cytotoxic one, has also CD8. What is a CD? CD means the cluster of differentiation, and these CDs are really co-receptors. They are helping TCR or T cell receptor in a signaling pathways, so they work together. Yeah, they are not just basic markers, and we don't use them just in pathology to marker specific cells, but they also help in the signaling pathways, so they are co-receptors. Remember that. And cytotoxic T lymphocyte had TCR, CD3 and CD8, and cytotoxic T lymphocyte is controlling or communicating with a cell which has an MHC class 1 receptor, main histocompatibility complex 1, which is showing antigen on this receptor, this cell over here, which is very happy to show something. And this antigen on the receptor MHC class 1 comes from proteasome digestion. As you know, cells have some kind of organelles to digest things, and every cell, almost every cell, has proteasomes, very special organelles that digest things, and in a way of proteasome digestion we create an antigen that is on the surface of MHC class 1 receptor, and this cytotoxic T lymphocyte is connecting with this MHC class 1. Everything is like close together, communicating and signaling. And when something is wrong with this antigen, because cell right here might be sick, containing viruses inside or something very dangerous inside the cytoplasm, when there are very, very dangerous antigens over here, the cytotoxic T lymphocyte will destroy this cell. So you really need a quite good antigen and quite good receptor, MHC class 1 receptor, for the cell to live. if something wrong here, the cytotoxic T lymphocyte will destroy this cell. On the other hand, we have helper T lymphocyte, which is only helping, isn't destroying anything. It's only helping in a reaction. Also containing TCR receptor, T cell receptor, CD3, because all T lymphocytes have CD3, but this time CD4. So it's CD4 positive, it's CD4 positive, CD8 positive. This helper T lymphocyte is communicating with a cell that contains MHC class 2 receptor. Oops, MHC class 2. This cell with MHC class 2 is also presenting antigen on its surface. And what cells do contain MHC class 2 receptor? These are not normal cells like these cells, but these cells that contain MHC class 2 receptor are antigen presenting cells. Yeah, antigen presenting cells contain MHC class 2 and are presenting antigens that they have found somewhere, and these antigens comes from lysosomal digestion. So lysosomes will create this antigen on the surface of antigen presenting cells. So this is very much what is happening in your body. Also, we have some natural killer T lymphocytes, which are very important to cytotoxic T lymphocytes, but they are part of adaptive immunity, and regulatory lymphocytes, which are regulating these immune responses. So the cytotoxic and helper T lymphocytes are very important. Also, from these CDs, you should know at least that B lymphocyte contain higher number CDs, so it's 19 and 20, and also they do contain a B cell receptor on their surfaces, B lymphocytes. So this is important. I'm sorry, this one, and also these right here, CD8. You should know at least these things right here, okay? As I told you before, here on this picture we have B lymphocytes, which will eventually turn into plasma cells, which will produce antibodies. B lymphocytes can bind to any antigen, but not so specifically. You want a specific immune reaction. So what will B lymphocyte do? It will divide into several types, and the best one will produce very specific antibodies. There are some kind of mutations going on here, so each B lymphocyte is slightly different, producing slightly different antibodies, and the best one wins and will produce antibodies that are very specific for the antigen right here. So effector cells here are plasma cells. These are effector cells because they are actually doing something. They are producing something, which are antibodies right here. For a T lymphocyte, effector cell of T lymphocyte is cytotoxic T lymphocyte. Cytotoxic one because it's producing enzymes like perforins and granzymes. Perforins are special enzymes that are producing holes in the cell. So here we have a nice cell with, as you know, MHC class 1 that is somehow disrupted or damaged, and cytotoxic T lymphocyte will produce perforins, and these perforins will make holes to this cell, which is now very sad because it will, I'm sorry, my pen is broken, but you can see it's very sad, tears, because it will undergo destruction right here. So because it will have holes inside the membrane, which is very tragic for the cell. And what's going to happen next? Well, we have this picture from loose connective tissue right here. So we have nice fibroblasts over here, spindle-shaped cells. This is collagen all around, collagen fibers, and these are the plasma cells. Plasma cells are once again effector cells of B lymphocytes containing clock shaped nucleus. Because of the organization of heterochromatin here with the basophilic cytoplasm, because plasma cells contain a lot of rough endoplasmic reticulum. This is once again also the fibroblast over here, and what we can see is something else I don't really know. Normal plasma cell. Very important is this picture from electron microscope, because this is a part of a cytoplasm of a plasma cell, and you should know this. Because how? Because there is a mitochondria, okay, but mitochondria can be found in all cells, most of the cells of human body, so that's nothing special here. But what is very special is this rough endoplasmic reticulum over here, which is very white. It's very white, and it's because plasma cell is producing immunoglobulins, which are stored here. So we have immunoglobulins inside the rough endoplasmic reticulum, and this plasma cell will produce the antibodies, store them inside the endoplasmic reticulum, and will wait for stimulation. So that's why the endoplasmic reticulum, rough endoplasmic, because there are ribosomes attached to it, it's so white because of the immunoglobulins that are stored here, the antibodies. And this plasma cell is waiting. This is very typical feature for plasma cell. So you should recognize plasma cell in the electron microscope just based on this feature. This is very special for a plasma cell. So we don't have only one type of helper T lymphocytes, we differentiate many types of T lymphocytes. And each T helper lymphocyte right here, this is the T helper type 1, 2, and 17. These are very important, these three types. Each type will stimulate a different reaction. So for example, TH1 will activate macrophages. TH1 will produce some signals, or the molecules that work as signals that, for example, could be like interleukins and interferons right here. So TH1 will activate macrophages because of the production of interferon gamma. So TH1 will produce interferon gamma, which will eventually activate macrophages to phagocytose something. TH2 will produce interleukin 4 and 3, and that will activate eosinophils and basophils, because there are some kind of parasites here in our body, so we need to fight a parasitic infection. These activations are important because they compete with each other. So when there are TH2 lymphocytes, when they are activated, this pathway of TH1 lymphocyte is deactivated. You will learn more about this in immunology. Right now, for the histology, you should at least know there are some types of helper T lymphocytes, and that will be pretty much okay. So once again, to antigen presenting cells. As you know, these cells have very special receptor, that is a MHC class 2. We can find this receptor only on antigen presenting cells right here, so this is very special for them. Of course, at normal cells and all cells in human body, except erythrocytes and except other types of cells that we are going to learn more about in next lectures, all cells should have also MHC class 1. This is just basic normal receptors that is located on every cell, and also antigen presenting cells have this receptor, but they do contain MHC class 2 receptor that is very, very special for antigen presenting cells. And these cells really don't do anything that is like in terms of production something, or this was very hard sentence because I'm sorry, like they don't produce anything. They are really important just for signalization. And we differentiate many types of antigen presenting cells like dendritic cells, Langerhans cells that can be found in the skin, in epidermis, follicular dendritic cells that are in follicles, in the lymph nodes, M cells that can be found in the gastrointestinal tract. Macrophages also are antigen presenting cells, because you know macrophages are in normal tissue, like they're everywhere, almost everywhere, and they are eating everything around them, so they are presenting these antigens to immune cells to like show them, okay, is this okay or it's not okay? What do you think? Because macrophages really don't know for the first time when they see some kind of cell and they eat the cell and now they present antigen, and T helper cell will help them because they are called helper cells because really they are helping other immune cells with the immune response. So T helper cell would tell macrophages like to, oh, that's not very good antigen right here, so you should do something with it. Okay, and that's a stimulation of immune response. So also some clones of B lymphocytes are antigen presenting cells. So also B lymphocytes, some of them can also phagocytose or eat something, but you should know these types, all of them. You should know all of them, and when they are located, we are going to learn more about them in the next lectures. Well, this is a picture of Langerhans cell. It's labeled with L, Langerhans cell. This is really epidermis right here, epidermis. And Langerhans cells are antigen presenting cells in epidermis right here. This is some kind of special staining, so you know you can see the processes right here. They are very big cells, with very long processes that are around cells that are normally located in the epidermis right here with this special staining. But it is the normal picture in hematoxylin eosin stain. They are very light cytoplasm, light active nucleus, and they are immune cells here in the epidermis. This is a picture from electron microscope. You can see that the nucleus isn't quite spherical. There is some kind of indentation. There is normal, like there are things around here, other things. I can really tell, probably some parts of epithelial cells around. It's hard to tell what is everything around here. Probably some epithelial cells around because we're in the epidermis. And what is here? This is a structure that looks like a tennis racket here. It's called the Bag Granule. Oh, sorry, Bag Granule. We really don't know what is composed of this Bag Granule. Probably some proteins that are needed for immune responses, but they are very typical for Langerhans cells, these Bag granules. So once again, back to classification. So that was like immune response or immune system in general. So don't forget about innate and adaptive immunity and basic components of these two immunities. And now let's look at the lymphoid organs and their histological structure. So once again, we have thymus and bone marrow as central lymphoid organs, and lymph nodes, spleen, tonsils as peripheral lymphoid organs. First, let's start with lymph nodes. So the lymph really must pass through at least one lymph node before returning to the venous system, as I said before. So the lymph enters the lymph node via these afferent lymphatic vessels over here. And afferent lymphatic vessels here, which enter through the capsule that is around the lymph node. So this is a normal picture of one nice lymph node over here. And as you can see, lymph nodes are encapsulated organs. So capsule is all around the lymph node over here. And it's merging, this capsule is merging these things over here. And these orange things are trabecules. That's something different as a septa. So they are not, that's not a septum. It isn't a septum, it's a trabecule. It's something like a column. So it's not like the whole wall as a septum. It's not dividing a lymph node into compartments or anything. It's just for the support of lymph node, like the columns that are going down to the center of the lymph node from the capsule. So nothing special really in the lymph node. So lymph goes from these efferent lymphatic vessels here to the lymph node. Here in the lymph node we can find sinuses, lymphatic sinuses here. There are many types of lymphatic sinuses, so just write it down. Sinuses here. It's just like open system. There are just like open spaces in the lymph node, and then the lymph goes back outside from the lymph node via efferent lymphatic vessel over here. The mostly only one, but there can be 2 of them, 3 of them, but mostly one efferent lymphatic vessel is going outside of the lymph node. So once again, afferent lymphatics, many of them going through the capsule to the lymph node, to the sinuses over here, and then going outside of an efferent lymphatic vessel over here. So the lymph is going like this way, from the periphery to the center, where we can find this place over here, and this is a hilum or hilus. In the hilus or the hilum we can find this efferent lymphatic vessel, and also we can find artery and vein there, because also lymph node needs some kind of blood supply. Yeah, it's a normal organ, needs a blood supply. It cannot live of like the lymph that is inside. It needs some oxygenated blood normally. So also we can find these big vessels over here in the hilum. So we can find many structures here in the lymph node, and as I told you before, the sinuses, they are very important. Sinuses, just like normal spaces here in the lymph node. I'm going to show you some of them. And also lymph is divided into 3 basic types that we are going to describe. So I think that's everything I wanted to tell you in this picture right now. And let's go here to another picture. This is a better picture, I guess. This is just also normal picture here, and once again what we can see right here, it's a capsule all around. This is a capsule. Normally when you find a capsule that is encapsulating some organ, it's made of dense irregular connective tissue. Okay, so dense irregular connective tissue is not very good short name for the dense irregular connective tissue, but never mind. And also trabecules are merging down from this capsule to support the structure of the lymph node. We divide lymph nodes into 3 basic parts, and that is a cortex, the first part. Cortex, it's on the periphery, of course, and it contains follicles. So when you find a follicle, you're in the cortex. I can tell you, you're in the cortex. Next, we have a paracortical region, which should be somewhere over here. Paracortical region. Paracortex. I don't know if it's written somewhere here, probably not. So the second one will be paracortex. That's the second region over here. And here in the center, we can find the medulla. Medulla in the center. Third part. So actually 3 basic parts. You should know them, you should be able to describe them. So as I told you, cortex contains follicles. Here then we have a paracortex right under the cortex, and then we have a medulla. There are some specific things that are inside each part, and we're going to talk about them. Yeah. Lymph nodes can be enlarged, and sometimes you can feel them really on the neck and or somewhere else, but don't worry, that's pretty normal. Probably you're just finding some kind of infection that you even don't know, so it's not a tumor probably. Okay, so once again, this is a histological picture of one lymph node over here, which has a capsule around. And these structures, these round structures over here, are lymphatic follicles. So these are lymphatic follicles. So this is the cortex on the periphery. Sometimes it's so curved we cannot tell really and differentiate medulla and cortex. Sometimes because of the section we're not even able to see medulla, and it's pretty normal. But this time we can see medulla. It's in the center, this light part. We can see it's very light because it contains sinuses that are in the medulla and that are called medullary sinuses that are very white. So that's why it's so light. So this is a medulla in the center, and as you already know, between medulla and cortex there is like a part called paracortex between them. Okay, supporting structure of lymph node, as you remember, I hope so, is reticular connective tissue. So reticular connective tissue is a supporting structure. And what is really a reticular connective tissue? These are the reticular cells together with reticular fibers. This is a special staining. This is not how it looks like normally. It's impregnation, and that's why we can see reticular fibers very dark. They are almost black here in this staining. It's impregnation, and these are reticular fibers over here, like these. These reticular fibers are produced via reticular cells that are in here. Reticular fibers are collagen type 3 fibers, so it's normal collagen, but just a type 3. Here there is an arrow pointing to a reticular cell producing these reticular fibers over here, and we have normal lymphatic cells all around. So okay, you can see right here the structure. You're not able to see this structure, reticular connective tissue, in normal staining, but you can see it very well in impregnation. So as I told you, we differentiate many sinuses and lymph nodes, and what's happening, afferent lymphatic vessels are protruding to the lymph node from the periphery. On this picture we can see really afferent lymphatic vessel going inside the lymph node right here. It's a normal vessel, it's lined with endothelial cells right here, so this vessel over here. And this, it's labeled with B, is a valve over here that can be found in lymphatic vessels, and they are more frequent, as I told you before. So normal endothelial cells that line this afferent lymphatic vessel. So this structure over here, what is this all around, this afferent lymphatic vessel? We know we are on the periphery, so this is the cortex. We're not able to see follicles, I know, but we know we are in the cortex when we see afferent lymphatic vessel going inside. This is in impregnation once again, so we can see nice black reticular fibers over here that are impregnated, and also there are many reticular fibers here and around this lymphatic vessel, and also in the capsule, which is on the periphery right here. So what's really with the sinuses inside the lymph node? When we go back to previous picture over here, as I told you, afferent lymphatic vessels, which we saw before, right now the afferent lymphatic vessels go through the capsule here, and they have normal endothelial lining over here. So normal endothelial lining over here, and the lymph, which is inside these lymphatic vessels, is poured into the sinuses that are located inside the lymph node. And we differentiate many types of sinuses. First type, the biggest sinus over here, which is really close to the capsule, it's under the capsule right here, it's called Subcapsular Sinus. It's a very big sinus containing lymph over here. It's not lined with anything, it's just the open space that contains the lymph inside. Then we differentiate intermediate sinuses, or sometimes called parafollicular sinuses. Parafollicular sinuses, because they are close to follicles, so that's why they call parafollicular sinuses. And the last type are medullary sinuses. Medullary sinuses are of course in the medulla, and they are white, they are the biggest. Not so biggest, because really the subcapsular sinus is very big, so probably they're bigger than normal sinuses in cortex right here, medullary sinuses. Once again, we don't have follicles in medulla, and that's how we differentiate medulla from the cortex. So back here, afferent lymphatic vessel that's lined with endothelial cells like normal vessels. And then what we have here in this picture, this is a capsule made of dense irregular connective tissue, like normal capsules are made. And this space here, it's subcapsular sinus over here. It's just empty space that isn't lined with anything. There is just reticular connective tissue around. Yeah, so these are normal reticular fibers over here, which are supporting the subcapsular sinus, but it's just open space. So this is the subcapsular sinus, this is the cortex over here. Here we have once again subcapsular sinus. This is the capsule over here, it's labeled with A. Then we have special cells here, like reticular cells over here. We can see their nuclei and also some processes over here. Reticular cell producing reticular fibers. And this is the cortex once again. So this is a picture from electron microscope, which is showing us how reticular cells really look like. So normal active nucleus, a lot of mitochondria over here, and also rough endoplasmic reticulum over here. And here what we can see are fibrils of collagen type 3, which are together creating a reticular fiber. So we can see individual fibrils over here, these small electron dense dots. We're not able to see fibrils in light microscope, we can only see fibers. This is a fiber. Yeah, these are the fibers. And this is really how the structure looks like. As you can see, it's more of a network of reticular fibers over here. And here we can see nice cells, probably lymphocytes over here, sitting there, doing their job. So reticular fibers are creating this network. Once again, a capsule, and subcapsular sinus. Subcapsular sinus, because it's under capsule, just a space. Sometimes you're not able to see subcapsular sinus in some specimens or slides, so don't be scared. But sometimes you can see it pretty well. Cortex. And once again, you can see the capsule, which is going to the trabecule right here, like the columns supporting structure, and this subcapsular sinus. Subcapsular sinuses are eventually going to another type of sinuses, which are connected together, of course, and this space here, just space filled with lymph, right here is parafollicular sinus or intermediate sinus. I know that because I can see a nice follicle over here. This is a follicle. So this is parafollicular sinus. Here we are in the medulla. I will tell you we are in the medulla because these spaces over here are dilated, and these spaces are medullary sinuses. And they will eventually merge into one efferent lymphatic vessel, which is once again lined with endothelial cells. So we have like a tube, efferent lymphatic vessel, normally lined with endothelial cell. Then we have a lymph node, which contains sinuses, just spaces inside. And then once again we have a tube, which is lined with endothelial cells, and it's efferent lymphatic vessel over here. Medullary sinus, this one. So this is the hilum of lymph node, where we can find efferent lymphatic vessel, which is very similar to vein. Sometimes it's very hard to tell, this is the vein, this is efferent lymphatic vessel. Both of them have very thin wall, and we can see a valve here, right here, very nice. And what else can we find in hilum? Of course, branches of arteries here and veins also, but we can find loose connective tissue here, and also many adipocytes are here. As you can probably see, many adipocytes over here. So adipose tissues everywhere and loose connective tissue together with these big vessels here in the hilum. Nothing special that much. So once again, this is a very nice picture of a lymph node. We can see a capsule made of dense connective tissue, irregular dense connective tissue. Here is the subcapsular sinus. Probably there should be some parafollicular sinuses, these white spaces over here, and medullary sinuses that can be found here in medulla. Cortex contains these lymphatic follicles over here, right here. And now we have a closer look on lymphatic follicles, what is really going inside the lymphatic follicles right here. We differentiate primary and secondary lymphatic follicles. Secondary lymphatic follicles contain something called germinal center. So secondary follicles contain germinal center, and there is this light spot in the center of a follicle, like here. Something going on here, germinal center. And in germinal center, there are many B lymphocytes that are very active, that are proliferating. So here, these B lymphocytes got in a contact with antigens. So when you have antigens here in these follicles, there will be also germinal centers here. Primary lymphatic follicles, don't contain germinal centers. They are dark basophilic with no lighter basophilic center inside them. these primarily follicles, really, we cannot find them in our body because all lymph nodes got into contact with antigens in some kind of way. So how does it look like really? This is, for example, the follicle, and here we have a germinal center in the center. So sometimes we will do a section through a follicle, and sometimes we are not really able to see the germinal center. But it's not because of that germinal center doesn't exist in that follicle. It's because of the section. So sometimes we can just see these dark circles over here with no germinal center inside, and that's only because of the section. Probably there is some kind of germinal center inside because all follicles got into contact with antigen in some kind of way. So what is really happening here? So this is the picture of secondary lymphatic follicle. It's a secondary follicle because we can see a nice germinal center in the center of the follicle, and around we have mental zone. So this is the mental zone all around germinal center, and around mental zone we have a zone on the periphery of the follicle that is called marginal zone. It's not written here, but you can see it's all around this follicle. This zone around is called marginal zone. Marginal zone. Follicles are really B lymphocytes clumped together into this round structure. So many B lymphocytes are here. But in addition to B lymphocytes, there are also follicular dendritic cells. Yeah, dendritic cells in the follicle are called follicular dendritic cells, and they are antigen presenting cells here. So they are presenting antigens in this region. Also we can find T lymphocytes here, T helper lymphocytes also in the follicle, but they are mainly around the follicles over here in the interfollicular zone, which is also the T zone around the follicles, but some of them are also in the follicle. There are many macrophages here, macrophages, because a lot of cells are dying here in this follicle because they are not good enough, so they die. And macrophages eat them up, so they phagocytose dead cells over here. And also antigens, because macrophages are also antigen presenting cells. Okay, so that's what's happening here right now. The result of this effort, because in a germinal center, what is really happening, B lymphocytes are proliferating, and there are many mutations going inside their DNA, and they are proliferating and try to create the best cell ever that has very specific antibodies for antigens. So what is the result of this is that the B lymphocyte that will go to this lymphatic follicle, B lymphocyte will go there, and the result of this process is plasma cell. Plasma cell is he over here, which is producing antibodies. So please, if you really don't know how to describe the follicle, at least during oral examination, you should be able to tell that B lymphocytes in the follicle undergo maturation, differentiation, and they form a plasma cell, which is effector cell of B lymphocyte, producing specific antibodies. But you should know many other things, and I'm going to describe them to you right now, what is really happening here. In this picture, once again, impregnation. We can see nice dark reticular fibers over here, and that's normal stroma, or the normal supporting structure of lymph node, as you know. So how does it actually work? The B lymphocyte arrives in the bone marrow. B lymphocyte is created in the bone marrow. It travels to the node, the lymph node, where it encounters an antigen. So there is an antigen in the lymph node, and this antigen is presented by antigen presenting cells. These B cells here, as I told you, wants to become a plasma cell that will produce antibodies which are very specific for this antigen. Thus, these cells, these B lymphocytes, began to multiply. They proliferate and they mutate. They are recombining genes. There is a recombination of genes going on here, and they create several versions of themselves. And when they start to proliferate, they are very active and they are called centroblasts. So these B lymphocytes will eventually become centroblasts. Centroblasts are very active cells. They are very big with a light nucleus inside. There are many of them because they are proliferating, dividing very well. And they create something called a dark zone over here. So this is the whole structure over here is a germinal center inside the secondary follicle over here. And these centroblasts are in something called a dark zone. It's labeled with DZ over here. And although it's called a dark zone, it's called the dark because there is high concentration of cells here. It should be called more dense zone, not dark. But these cells are light. They are lighter than cells that are located in a light zone. I know it's very confusing, so just focus right now. B lymphocytes proliferate, they create centroblasts, which are very active cells. They are very light, very big, and they are proliferating. There are many of them, and together they create a dark zone in a germinal center. After that, many of them die because they are not good enough. Many of them die, and some of them become another level, and that is I'm sorry, sometimes this happens with my pen, I don't know why, but centroblasts will eventually become centrocytes. Centrocytes are darker cells, and they do not proliferate, and they are located in a germinal center in a zone called light zone over here, light zone. And they will eventually differentiate. Not all of them. Some of them will die, of course, once again, but some of them will differentiate into plasmablast. And plasmablast will differentiate into plasma cell producing antibodies. Or there could be another pathway that could enter. Centrocyte could differentiate into memory cell. Memory cell is something similar to plasma cell, although it isn't producing antibodies right now, but it can be activated and become a plasma cell and produce antigens once again when it will meet antigen somewhere in an environment, this memory B cell. Okay, so B lymphocytes turn into centroblasts, then centrocytes that are in the light zone in the germinal center, and they will turn into plasma cells or memory cells. It really depends on the stimulation. Once again, we have it right here, a dark zone which contains centroblasts. There are many of them, and the light zone with lower amount of centrocytes because many centroblasts died here in the dark zone. And these centrocytes will either undergo apoptosis, once again because they are not good enough, or they become a memory cell, or they become a plasma cell. Okay, so you should know at least these like basic types of cells here in the germinal center. Once again, it's written here. Remember these follicular dendritic cells, which are antigen presenting cells over here, And also we can find T helper lymphocytes over here, helping with the interactions over there and stimulation. Here we have once again a picture of a lymph node. There is a nice capsule around, and there are no septae but trabecules, as you know. A supporting structure is made of reticular fibers and reticular cells. This is an hilum over here. And why is this picture important? Because we can really see the paracortex here, the layer, the region between cortex and medulla. Cortex contains follicles. Follicles contain mostly B lymphocytes, but there are also T lymphocytes there. Don't forget that. And also T lymphocytes are around follicles in the cortex. And paracortical region is important because it contains many T lymphocytes here. And paracortex has something called high endothelial venules that are very important for paracortex, because T lymphocytes will enter lymph nodes here in this through these venules. What do I mean by that? Is that you have many T lymphocytes normally in your blood circulation, and when T lymphocyte will enter lymph node, they are entering via these high endothelial venules in the paracortical region. These high endothelial venules are specialized for transportation of T lymphocytes here. The endothelial cells are very high, they are cuboidal here, and it's kind of paradox, but they can transport T lymphocytes better than normal capillaries or normal vessels here. So, and this is a medulla normally. And next we have a nice picture from electron microscope. This is an electron microscope of a high endothelial venule over here. So these are endothelial cells, and they look very different than normal endothelial cells. You would say that is some kind of duct lined with normal epithelial tissue, like epithelial, simple cuboidal or simple columnar epithelium, but this is really a venule right here, and it's very hard to recognize it. So don't worry about it. This is just for you to see. You don't have to recognize it really, but you should know they are venules with high endothelium in the paracortical region, and their function is to transport T lymphocytes here in this region. And how are T lymphocytes transported? You already know two main processes, or diapedesis. Diapedesis, it's a transportation of leukocytes between the connections between endothelial cells. And when you want to transport immune cell through cytoplasm of endothelial cell, is called emperipoiesis. I don't know how to pronounce that correctly, I'm sorry, but yes, this is processed through the cell. So you should know that we can transport cells between endothelial cells and through endothelial cells, of course. Well, wow, this is a very good picture. First, I really didn't know like what's going on here in this picture, but this is a T lymphocyte, probably. Okay, so we cannot tell, but this is a T lymphocyte right here that is communicating, or it's like really attached to the process of dendritic cell. So this is probably one process of dendritic cell over here, like antigen presenting cell, and they are almost attached to each other, and there is some kind of stimulation going on. So remember, there are receptors on T cell, and there are many receptors here on antigen presenting cell, and they are stimulating each other. And this is how it looks like. You will learn more about this in immunology, I'm sorry, because there are many other receptors than CD4, 3A and other stuff, but it's not so important right now. But it's more complicated than as I explained before. This is a lymph node from our department. This is a picture of our slide of lymph node. So you can see lymphatic follicles on the periphery right here. In some of them you can see the germinal center right here. Some of them don't contain germinal center, but it's really because of the section, as I told you before. Probably there is some kind of germinal center somewhere there, but we're not able to see it. Once again, also here. So this is the cortex, because I can see many lymphatic follicles over here. I can say this is the medulla, because it's more lighter region over here, medulla. And probably somewhere around will be a paracortical region. It's not so important to really tell like the borders between, just to be like familiar with the terms, and that the cortex is on the periphery and medulla is in the center. Yeah, so nice follicles over here. And between follicles there is this region that contains also some lymphocytes, mostly T lymphocytes and B lymphocytes are here in the follicles. You can see there is some connective tissue around. There are many adipocytes here. That's pretty normal. Location of lymph nodes is very different. Some of them are more in adipose tissue, some of them are not. So it depends, really. So here we have a nice germinal center over here, and I can see many cells that are light are located here on this side. So probably I will tell that this thing right here, this is the dark zone that contains many cells that are centroblasts, and this is the light zone that contain less cells here in this picture. And here we can find astrocytes, probably more differentiated B lymphocytes, and all around we are probably able to find many plasma cells here that are fully differentiated, able to produce immunoglobulins. And once again over here, so germinal center, mental zone around, darker zone, and marginal zone on the periphery, marginal zone. Well, and the spleen. Spleen is another peripheral lymphoid organ which is fully encapsulated and is very big and is very important. I can tell you, it's like you can live without spleen, but you rather have spleen. It's very important. It's the largest lymphatic organ, and spleen filters blood. It's not like in a lymph node, that lymph node, it's more of a it's more about filtration of the lymph, the interstitial fluid that is going in the lymphatic vessels, but spleen filters blood. It really reacts to antigens in the blood, and enlargement of the spleen is called splenomegaly. It can happen in some kind of infections and also tumor, in some kind of leukemias and lymphomas. It can enlarge. It's capsulated, okay? And basic stroma or supporting structure of spleen is once again reticular connective tissue. So it's pretty much the same as for the lymph node. Yeah, reticular connective tissue capsule, and this capsule creates trabecules, once again, these trabecules of connective tissue. But this time, these trabecules that are merging from the hilum, this time it's opposite. Like for the lymph node, they were coming from the capsule on the periphery, but in the spleen, they're going outside of the hilum here. These trabecules are more important than in the lymph node. In the lymph node, there were some kind of just supporting structure, but in the spleen, they actually contain vessels. Trabecules contain vessels, and they are very important in the nutrition and the vessels that are inside them. I hope you understand what I'm getting to. So let's start with the basic description of a spleen. So this is just a picture of a spleen, and you can see there is a capsule made of dense irregular connective tissue that is going here in trabecules. These are very small trabecules. Sometimes we can see only some partial sections. Here is a very big trabecule, and you can see right now this trabecule contains big vessel over here right now, and also here. So, and this is the basic structure of a spleen, and supporting structure is a reticular connective tissue once again. So I want to go back here in this picture because you should be able to tell that the artery, lienal artery, is from the celiac trunk, and the vein that is going outside of the spleen is going to the portal vein and then to the liver. It's not here, but then it goes to the liver. So you should know the nutrition of the spleen here. So spleen divides into two basic parts. They are very important, and you should be able to describe and differentiate them. The first part is white. I'm going to put another color here, for example blue, and that is the first part is a white pulp, but not bulb, pulp. Pulp, white pulp. And white pulp basically is the lymphatic tissue over here, and we can see some lymphatic tissue over here. So this is the white pulp. And you can see it's all around some kind of arteries that are here. I'm going to describe them later, but you can see the lymphatic tissues around some vessels here in this spleen and in every spleen. And the next one is, second one, red pulp. And red pulp is made of cords and sinuses, venous sinuses. And it's in between the white pulp. So everything around lymphatic tissue here in the spleen is red pulp over here. You can see it's more like lighter in here in this picture. So it's all around this lymphatic tissue. So these are two basic components of spleen: white pulp and red pulp. This is a picture from our spleen in our department, what we have here. So this is a nice capsule right here, and we can see trabecules over here, which are made of dense irregular connective tissue such as a capsule around, and they are branching from the hilum, which we cannot see here because we are more in the periphery. And these trabecules that are inside here, once again, they are not dividing spleen into some lobules. No, it's not happening here. Trabecules are just like columns going inside the spleen, and they contain trabecular arteries and veins. They are called trabecular arteries and veins because they are inside the trabecules, so nothing special about that. Once again, on this picture, we can see white pulp over here. This is the white pulp. So basically lymphatic tissue over here. Lymphatic tissue. And all around the white pulp we can see red pulp. Red pulp is made of cords, I'm going to describe them, and venous sinuses that can be seen also in the small magnification, because these spaces over here are venous sinuses, these white spaces that contain blood. We're going to describe them, are venous sinuses. This is a picture of a capsule right here, so normal capsule. Okay, this is just some kind of section, small section of a trabecule right here. It's pretty much okay. Sometimes we can see just like this small trabecule. Sometimes we can see bigger trabecules containing vessels. So it depends, and this is just a schema of a white pulp and red pulp, what is in round. So we have lienal artery here from the cilia trunk over here. Lienal artery that is branching. It's branching in the hilum, so it goes to hilum. And we have these trabecules merging from a hilum of a spleen, and these trabecules contain branches of lienal artery, and these are trabecular arteries over here. So they are located in trabecules. They branch into another type of artery, and that is a central artery. Central artery, important, very important. And it's a very small artery. It should be called arteriole more, because it's very, very small, so it's not an artery, but it's called an artery, so we're going with that, like, okay, it's an arteriole, but doesn't matter. Okay, let's call it central artery. And all around central artery there is a white pulp. So we can see it's also in the slide that there is a central artery, and around we have some kind of lymphatic tissue, and that is a white pulp. White pulp has many I shouldn't say layers, but components, because right next to the central artery we can see some lymphatic tissue that is made of T lymphocytes over here right next to the central artery, and it's called PALS, like pulse. It means periarterial lymphatic sheath. Periarterial because it's around artery or arterial, lymphatic sheath, pulse. You should know this name, and you should know that it's time dependent because it contains T lymphocytes, many of them. So T lymphocytes are around central artery. Then we have another component of white pulp, and that is a peripheral white pulp. So PWE, peripheral white pulp, that contains mostly B lymphocytes, and sometimes we can also see follicles there, normal follicles. And you now know that follicles are made of B lymphocytes, so that's pretty normal. Around peripheral white pulp, which is composed of B lymphocytes with these follicles, we can see marginal zone around here. So there is also marginal zone here, which contain very important marginal zone sinuses, and there are very big macrophages here, and they are specialized here in immune response. So you should know that here we can see many macrophages in this marginal zone, macrophages. Well, but let's go somewhere else from here. The central artery is branching. It's branching and is creating some other arteries here, which are called peninsular arterioles. So it's branching into peninsular arterioles, which are very small. Sometimes we can see them, sometimes it's harder to see them, but these peninsular arterioles branched into another arterioles over here. It's not written there, but they branch into encapsulated or capsulated arterioles. Capsulated arterioles. Okay. And capsulated, they're called capsulated because they have a sheath around them made of macrophages. So, but what I'm heading to is that maybe I will use another picture. Right now, just remember the central artery is branching into small and smaller arteries, and there are many venous sinuses in a red pulp. So this is the white pulp over here, but now we're heading to the red pulp. So I will do this section over here. This is the red pulp, and here we have this lymphatic tissue over here. This is the white pulp. And red pulp contains a lot of sinuses and capillaries. And we somehow need to get this blood from central artery. We need to get this blood to the venous sinuses over here and to the venous system and trabecular vein that is inside the trabecule and outside of the spleen into the portal system. So how are we going to do this? This is very complicated and important also, and there are many theories how it's working in the spleen. So now just let's describe the white pulp and red pulp once again here in this picture, and you should be able to describe it also during your practical trainings. So we don't see any trabecule here, but what we can see clearly is this structure over here, and this is the central artery. Yeah, central artery, and also right here. And around central artery, it's called central, but it's not completely centrally, as you can see the white pulp, because white pulp is this whole thing over here around it. And this white pulp is composed of pulse, that is peri-arterial lymphatic sheath or lymphoid sheath that is somehow this big all around this central arterial. So pulse, there are T lymphocytes inside, and around pulse we have PWP, peripheral white pulp, which is composed of B lymphocytes and contains follicles. So this is the follicle right here, but also we have B lymphocytes over here. And this follicle is the same as the follicle of the lymph node. Basically, all things that happen in lymph node are happening also here in this follicle. Centroblasts, centrocytes, and everything. We're creating plasma cells here. And T lymphocytes are helping here to create the plasma cells. So, here is also white pulp, white pulp, white pulp, here, and here, and this is red pulp. Red pulp is made of sinuses, so these spaces over here, these are venous sinuses over here. And you can see some tissue around here. Don't forget the supporting structure is reticular connective tissue. So we basically have reticular connective tissue here. But what else? These are called the cords of Billerud. And these cords of Billerud sounds very difficult, but these cords are just reticular connective tissue with immune cells like lymphocytes, but also there are some erythrocytes there, macrophages, granulocytes. So all components that you can find in blood, everything you think of, can be here in the spleen between reticular fibers, forming these cords of Billerud. I think I hope I explained that clearly. Because now let's have a look on circulation. Not quite, you can see higher magnification right here. So this is a central artery, this is a follicle over here, this whole structure is white pulp over here. It's hard to see in higher magnification the differences. And once again, here we can see a nice trabecule over here containing trabecular artery right here and vein. So we can see very nice big vessels over here. So what's really happening in the spleen? Voila! So here we have, of course, central artery over here with periarterial lymphoid sheath around. It's dividing into penicillate arteries. You should know this name. They are dividing in a sheathed or capsulated arterioles, which are encapsulated via macrophages that are sitting around. And now what's happening? There is this theory of open circulation and closed circulation. So in one theory of open circulation that is probably more that is probably mainly in humans, this is happening in our spleens, is that the blood from these capillaries is just poured into this reticular connective tissue. So you have blood in your capillaries and now it's just in the spleen, in the reticular connective tissue. It's one place in the human body that the blood is just spilled out in some kind of organ. It's just between reticular cells here with reticular fibers. Isn't that crazy? And then when there's some kind of immune response, red blood cells are destroyed here because there are many macrophages here, so they eat up all the red blood cells, and then the blood is going back to the venous sinuses here, venous sinuses, and back to the venous system. Pay attention to a difference between venous sinuses in the spleen, which are these structures right here, and sinuses in the lymph node. There is a huge difference. Sinuses in the lymph node are just spaces with nothing around them, just spaces. Sinuses in the spleen are covered with endothelium, so they are more like sinusoids. Yeah, sinusoids. You can say also sinusoids in the spleen. There's nothing different between sinuses and sinusoids. Well, maybe there is some kind of difference, and I will show you the difference. But this is open circulation, and maybe in some animals, I think, you can find this closed circulation when capillaries are going directly to the venous sinuses, but this is more often in a human. So we have nice pictures over here. So these are venous sinuses in the red pulp over here. They are lined with endothelial cells. They are more like sinusoids, and between these sinuses we can see some cells, and we can see these structures over here are cords. These are the cords of bilirubin. They are just reticular fibers with erythrocytes and everything what's inside blood together, forming cords. This is another picture. We can see nice pulse around the central artery over here. Okay, nothing special about this right here. Also, once again, the pulse and normal peripheral white blood around. Okay, so this isn't a very good picture, but we can see venous sinuses over here in higher magnification. These are the venous sinuses. And there are cords between them formed by everything you can find in blood in general. So this is happening in a closed circulation, capillaries directly to the venous sinuses. Open circulation, we have open capillaries, and then we have sinuses. Once again, venous sinuses over here. Another picture, not so good picture, but you can imagine how it looks like. This is impregnation once again, and we can see reticular fibers here that are also around venous sinuses right here. And we can see accumulation of reticular fibers around central arteries right here. Well then, and this is a very nice picture because now we can see venous sinus in much higher magnification. This is from scanning microscope, of course, and these endothelial cells here that line venous sinuses are so long. They are very long, and they have many very big spaces between them, and they are called with special name, and they are called staph cells. But they are just normal endothelial cells, little bit elongated with bigger spaces between. And here we can see like a neutrophilic granulocyte. The macrophages are also here, but you don't worry, you don't have to differentiate them based on scanning electron microscope. This is just for you to imagine how it really looks like. And these are the reticular cells over here. The process of reticular cells forming reticular fibers. Once again, another picture of staph cells, specialized endothelial cells here. So these are the processes, very long processes of endothelial cells right here. Very nice. Nice picture. A nice neutrophil waiting here and a macrophage that wants to go inside the venous sinus here. Yeah. So spleen is very important lymphoid organ, but don't forget it gets rid of erythrocytes. Old erythrocytes die here, or when there is some kind of complication in erythrocytes, they are not good enough, they also die in the spleen. Yes, this is very sad picture. I'm sorry. Next, the thymus. The thymus is different, and it's a central lymphoid organ. So it's a epithelial organ. Why epithelial? Because supporting structure isn't just the reticular connective tissue, but we have reticular epithelium. Very important reticular epithelium, because it's important for pathology, because carcinoma, that is a tumor of epithelial tissue, can develop in thymus because it's made of epithelium. As I hope you understand this, why is it so important. And there is also a keratinization that can go on in this epithelium here in thymus. We have 6 types of epithelial cells together. You don't have to differentiate them. There's no way how to differentiate them in normal histological staining that we are using in our department. But you should be able to differentiate cortex of the thymus and the medulla. We also differentiated cortex and medulla in lymph node, as you remember, hopefully. And in the spleen we have the white and red pulp, and in thymus once again we have a cortex and medulla. The function of the thymus you already know: development of immunocompetent T lymphocyte, which developed here. They develop immunocompetence here, of course. They come from bone marrow to the thymus, and thymus is evolving in time. So you have thymus. You had thymus when you were kids, but now after puberty, probably you don't have thymus anymore, but there is some kind of tissue there that was thymus before, so there are some kind of remnants that stayed there. So there isn't like that the whole thymus will disappear in time. It's no, but it's evolving in time. It's getting smaller and smaller. But there is still some kind of tissue there, and there are some kind of remnants of reticular epithelium there. It's of course behind the sternum. It's located in the superior mediastinum here, and it consists of 2 lobes, sometimes 3 lobes, and we can find septa there in the thymus. So in the lymph node and spleen we have trabecules. Now we are having septae, but they divide the thymic parenchyma in the pseudolobules. These septae divide thymus into pseudolobules because these septae do not go straight through the thymus. Everything is connected together in the thymus. So we are not describing lobules, separated lobules, but pseudolobules. Everything is connected. So this is how it looks like. This is the thymus, of course, with the capsule around. We always need some kind of capsule around something. So dense irregular connective tissue around, and we can see some kind of septae. But what is this? This is a lobule. Like right here, lobule, and lobule, another one. But you can see these lobules are connected with each other. And that's why they are called pseudolobules over here. We differentiate lighter medulla inside, in the center, and darker cortex. there are more T lymphocytes here in the cortex. That's why it's more basophilic. Medulla is lighter because there are less T lymphocytes, more matured ones. Many of them died here in the cortex. Some of them will die also in medulla, but they are more matured here in medulla. So each lobule, each pseudolobule has its own cortex and its own medulla that are connected together with another pseudolobule that is next to the first one. This is a higher magnification, so here we have a nice capsule around the thymus, and the lighter area over here is the medulla, and this is the cortex over here. Supporting structure once again are the reticular epithelial cells. So don't say just reticular cells because that's not true. These are reticular epithelial cells. This epithelia is very important. These are connected to each other with desmosomes. There are very tight connections between the protrusions of cell, the processes, and also we can find lymphocytes between them. Many lymphocytes, and we can basically find only T lymphocytes in the thymus. T lymphocytes here. So in this picture we can see nice cells of reticular epithelium. They are very light, they are very active, they are producing everything around the supporting structure, and they are connected with these processes here. This is a picture from electron microscope. These are of course lymphocytes around, and this is a epithelial cell because we can see bundles of tonofibrils, and you already know tonofibrils. These are intermediate filaments, cytokeratin, connected together and creating tonofibrils, like bundles of intermediate filaments over here. And this is a typical feature for epithelial cells, so you should know it. Tonofilaments, once again, these are the nuclei over here and also here, and desmosomes, connections between two epithelial cells right here. So this is a picture from our thymus in our department. Once again, there is a capsule on the periphery. These are the septae over here, which contain vessels, as you can see right here. So we can find nice vessels here, and are dividing the thymus into pseudolobules like this. So I will describe, this is some kind of, it really looks like in this case, this lobule, that it looks like it's separated completely, which is not very true, but we know that they are connected together, these pseudolobules here. And you can see very dark cortex in each lobule and light medulla in the center. Yeah, once again, medulla and cortex. You can see right here, you're not able to find very big vessels in the cortex, only these little things going here. These are the capillaries, small capillaries in the cortex. And these capillaries are very important because antigens are entering the thymus with these capillaries that are in the cortex right here. But you don't want to react on everything that's outside. So only some antigens can enter the thymus. So we have something called thymic blood barrier. So the barrier between blood and thymus. We have capillaries that have continuous lining. Yeah, so this is endothelial cell here, this nucleus, and continuous lining. So yes, and also epithelial cells, reticular epithelial cells have processes around the capillary to make the barrier even thicker. So reticular epithelial cells are all around the capillaries, making it even harder for antigens to enter the thymus. But what is really happening in the thymus? is that, as I told you before, we have many T lymphocytes here in the cortex and less T lymphocytes here in medulla. And what's happening? In the cortex, we differentiate something called positive selection. Positive selection. In medulla, we talk about negative selection. So their T lymphocytes are building their immunocompetence here in the cortex, and positive selection means that they are developing receptors, and if they do not develop receptor or they cannot bind to any antigen, they will die. Okay, so if they are not capable of producing any receptors on their surface, they will die. They undergo apoptosis, and that is what they call positive selection right here. So like 99% of T lymphocytes will die here in the cortex. It's very hard to build receptors, I can tell you. But if they do so, now we have in medulla, we have T lymphocytes that contain that T cell receptor, and it's quite good. But what can happen also in medulla is that this T cell receptor will bind to self-antigens, and we don't want that. We don't want some kind of autoimmunity or autoimmune reactions going on. So if the T lymphocyte will bind to self-antigens very tightly, they are very aggressive, starting to be aggressive to self-antigens, they will die. And that's what we call a negative selection. So positive, we know now, right now, negative also. So T lymphocytes are dying here, and some of them will persist, and I'm happy 99% of my T lymphocytes were dying here in the thymus, because I want really good T lymphocytes. So it's very good process, it's very hard process, and you should know how to describe positive and negative selection and what is going on in the thymus in general. So we have this barrier, it's very important, but also matured T lymphocytes needs to go away from the thymus. They don't want to stay here. So once again, as in the lymph nodes, once again here in the thymus we have venules, high endothelial venules, and we can see it. They are here somewhere in the border. Here, this is the medulla, and this is the cortex. It's more darker here in the cortex. So sometimes we can see high endothelial venules like this. This is the cross section over here. These are the endothelial cells, and T lymphocytes that are matured, that are very good at their work, are leaving thymus with these venules right here. They are leaving thymus this way, through venous with high endothelium. This is once again how it looks like. They are less numerous in thymus than in a lymph node. And yeah, you should know like why do we have this kind of venules in here. How do we differentiate the thymus once again, and the medulla and the cortex are these Hassel bodies. And what are Hassel bodies or corpuscles? These structures over here. Because I told you that this reticular epithelium undergoes keratinization, and reticular epithelial cells will die in some time, because thymus is evolving, so reticular epithelial cells start to die. They connect to each other and form these Hassel bodies over here. And they are more eosinophilic. You can see the eosinophilia. It's because of the keratinization that is going on inside. Sometimes calcium will... I'm sorry. I'm sorry. Sometimes calcium will go to this Hassel body, and there's sometimes some kind of basophilia here, it's because of the process called dystrophic calcification. Because when something is dying in your body, calcium really enjoys that. Something is damaged somehow, so calcium goes there. So when there are reticular epithelial cells dying, they're keratinizing, and it's happening really inside the body, calcium is like, oh well, party, I should go there. So sometimes we can see some kind of basophilia also here, and that's because of the calcium. And when we are older, we have bigger Hassel bodies and more of them. So it really is because of the involving of the thymus. The thymus is slowly dying. And here we can see nice capillary in the cortex over here. And these Hassel bodies are only in medulla. Yeah, only here in medulla. And that's how we can really differentiate medulla and also the thymus, the whole thymus. And this picture, I don't know why do I have it here right now, but maybe you can have a closer look on the Hassel body over here. Oh, and maybe these are all lymphocytes over here, yeah? But sometimes we can see light cells, more light, like these over here, and these are reticular epithelial cells over here. Sometimes we're able to see, maybe we can see it over here, the very strange cell over here, which has deep indentation in the nucleus, and you probably can guess right now it's a macrophage. So we can see many macrophages because many cells are dying here. It's pretty much okay to eat them. So many macrophages. Once again, a Hassel body over here. So accumulation of reticular epithelial cells together creates a Hassel body. Once again, Hassel body is over here in medulla. And this is a picture of electron microscope of a Hassel body. This is a keratinization. This is how keratinization will looks like over here. And this is part where we can see calcification right here inside, probably. And epithelial cell around here, reticular. So during examination, if somebody wants to show me epithelial cell of the thymus, reticular epithelial cell, just find the Hassel body, because Hassel body contains dying reticular epithelial cells. They are light active nucleus, okay, but you can show them Hassel bodies. So what are the basic differences between the lymph node and the thymus? Thymus is central lymphoid organ. Lymph node is peripheral one. Both are encapsulated. We can find septae in the thymus. We can find trabecules in the lymph node, also in the spleen. But the biggest difference is lymph node contains also B lymphocyte. That means that B lymphocytes are creating follicles. So can we find follicles in the thymus? I don't think so. No follicles in the thymus, babes. No follicles. So there are only T lymphocytes in the thymus, and we are not able to differentiate follicles here. This is the basic differences. And Hassel bodies. Hassel bodies in the thymus. Oh god, this is the best feature. So, and now let's have a look on the last part of my lecture. And that is the lymphoid tissue that it's not completely encapsulated, or so-called partially encapsulated lymphoid tissue, and uncapsulated one. You probably know what Walder's lymphatic ring means. This is a system of 6 tonsils in together. Actually, in our department we have pharyngeal. No, we don't have pharyngeal. We have palatine tonsil and lingual tonsil right now. But you should know how many tonsils do we have. Tonsils are, in general, the definition, they are partially encapsulated lymphoid organs. Don't forget that. And Walder's ring is composed of 6 tonsils, 1 pharyngeal one, 2 tubarian, 2 palatine tonsils, and 1 lingual tonsils. They have really protective function. They contain follicles, and also they contain T lymphocyte. So everything that happens in a lymph node It also is happening in the tonsils. So pretty much okay. Now let's have a look on the tonsils so I can describe the basic structure. Here we have a palatine tonsil, which is located here inside your mouth. You can see it if you have it. I don't have palatine tonsils, but probably you have them. So they are covered with epithelium that is normally inside the oral cavity, and they are covered, this palatine tonsil is covered with stratified squamous epithelium right here. But remember, it's partially encapsulated. So under the tonsil, tonsil, you can see it right here clearly, there is a capsule. Normally there's irregular connective tissue under, but only from the side where the tonsil sits here in this loose connective tissue with muscles and everything that is under the tonsil right here. So here we can find capsule. There are septae protruding from the capsule here to the periphery that contain vessels over here. And tonsil, it's really accumulation of lymphatic tissue, lymphoid tissue here under epithelium that is covering the tonsil from this peripheral side over here. Capsule is under the tonsil, and we can see many follicles over here. It really looks like a lymph node, no? It doesn't to you? Like normal lymph node. So there are B lymphocytes here, and also T lymphocytes around follicles and also inside follicles. So we can find everything that was in the lymph node. And tonsils, like the palatine tonsil, it's a typical feature for this palatine tonsil, is that it's creating crypts, these invaginations of epithelium right here that are covered with epithelium that is on the surface. That's normal stratified squamous epithelium, crypts that contain like sometimes pus and mucus and fluid and stuff like that. And you can see the crypts also from your view when you have a close look and you open your mouth. You can see the palatine tonsils, and you're probably able to see also these invaginations of palatine tonsils. And don't forget, when you have a nice look on your palatine tonsils or onto some patient, you're able to see the epithelium that is covering the tonsil, but you're not able to see the capsule that is under the tonsil right here. So in higher magnification we can see nice stratified squamous epithelium here, and this is a follicle over here. You can see the lymphatic tissue over here in loose connective tissue of the mucosa that is here. Yeah, so lymphatic tissue, and capsule should be somewhere here in this region. And there is this crypt, deep invagination. This is our specimen, by the way. So these are the crypts, and under epithelium we can see lymphatic tissue, and under lymphatic tissue, this thing over here, this is the capsule. This is a nice cross section of a crypt. So this is just a cross section of a crypt with stratified squamous epithelium. Around we have a nice lymphatic tissue, and around we have a capsule. Yeah, once again, nice capsule around under lymphatic tissue. Everything that happened before in the lymph node and the spleen also is happening in these follicles here in the tonsils. So the function is the same. Also we have the lingual tonsil, and lingual tonsil is smaller. There are lower crypts there, smaller amounts of lymphatic tissue, but is also covered with squamous stratified epithelium. Also is the same as for the palatine tonsil here. As when I go back for the pharyngeal tonsil over here, Pharyngeal tonsil is covered with pseudostratified epithelium with cilia that is also in airways. So, and also that is the same for Tiberic tonsil over here. So these two have a different epithelium than these two. But on our slides, we have palatine tonsil and lingual tonsil, and they are both covered with squamous stratified epithelium. So, and what is happening here? We got to the intestinal lumen over here because we have some lymphoid tissue also in the intestines, our intestines in the lumen, in the mucosa that is close to the lumen. And we differentiate, these are normal enterocytes, you know them probably, that are in the intestines. Enterocytes, you know them from duodenum, do you remember? Columnar cells with microvilli, the brush border here, they are eosinophilic with oval nuclei here. But between them we have M cells. Not everywhere, but in some specific place where we can find accumulation of lymphoid tissue, and that's in ileum. We have these Bayer patches. We're going to learn more about them in next lectures. But M cells are antigen presenting cells, and they are communicating with the outer space. They are providing interaction between antigens and a lymphatic tissue that is under this epithelium right here. Maybe we can have a nice picture. Well, this isn't a nice picture, but probably here. This is ileum, and ileum have some layers. These are the villi of ileum, and you can see in the mucosa there is this part. This is a small part of Bayer patch. I'm going to write it down. Bayer patch or Bayer's patch, something like this probably. And that is accumulation of lymphatic tissue in the mucosa and the submucosa of ileum. And above this region, we can, in this epithelium over here, which is a simple columnar, we can find M cells. And how do we differentiate them? It's really not so important to differentiate them, but I can tell you that this cell probably over here is M cell because there are lymphocytes that are very close to it. Yeah, so this is probably an M cell because it's providing interaction between antigens and lymphocytes that are located in this mucosa over here. Sometimes, and that's pretty normal, we can see individual follicles in the mucosa. You can sometimes see it, it's pretty normal. And this was very tough lecture, I think, but I hope you could understand everything. As I said, I'm a little bit confused right now because I'm talking for so long, and I hope you have a nice day, and don't hesitate to contact me if you have some problems with the presentations or lectures and you want to ask some questions. So bye bye.